Now presenting is Cora Lyon with the next generation of sequencing of extremophiles, determining the biological phenotype of Dinococcus Radiodurans.
Hello everyone, my name is Cora Lyon and I'm a student at the University of Alaska Anchorage. I'm working on this project along with Matthew Redlinger, Ralph Dagdag, Jason Berghead, Brandon Briggs, and Eric Bortz. We are all at the University of Alaska Anchorage. I'm quite excited to be here today to present to you all. So I'm going to start by defining some of the terms in my title. So next generation sequencing, the specific platform I'm using is Nanopore Technologies, specifically the MinION sequencer. Extremophiles are bacteria or archaea, but in my case bacteria, that live in extreme environments. This includes high salinity, high and low temperature, high and low pH, and for Dinococcus Radiodurans, high doses of radiation. And then the biological phenotype is a mixture of the genotype, the phenotype, the genome, the transcriptome, the proteome, and the metabolome. So the genome is made up of DNA. The transcriptome is the RNA that is made off the DNA, so you can tell how much of stuff is being produced. The proteins, an even better indicator of what is going on in the cell. And then the metabolome is the different metabolites such as calcium and sodium, so you can tell what the cell is using. So to start off of how I got into this project is I joined the UAA robotics team to compete in the Mars Society International Robot Challenge. And I was put in charge of the science team. And shortly after that, a paper came out in a national biology journal called The Ladder of Life Detection. And I've snipped out the part from the important table that really solidified why we should be using next generation sequencing for looking for life on Mars. And that's because first you look for habitability, and then as you climb up the ladder, with the top being on the far this side and the bottom being on the far this side, you get more and more sure that you've been able to find life. So while creating the scientific suite of tests for our rover, we wanted to cover different components, different rungs on the ladder, but we also wanted to see how far up on the ladder we could get. And the farthest stuff we could get with what we could do with our resources was under molecular and structures conferring function, the polymers that support information storage and transfer for terrain life, both DNA and RNA. So this is where the project started. But what it's gone to is it's brought me here to what does people living on Mars look like? And as the titles say, we will bring microbes with us, whether we want to or not. So people have microbiomes not only on your skin, but also in your lungs, in your gut, in your mouth, in your ears. There's a whole suite of microbes that live within us and on us that help us throughout our daily lives. So as we move to Mars, or the moon, or throughout the solar system, this can possibly affect the relationship between the microbes and our bodies. And then if there's microbes already in the soil, it could affect any plants we try to grow. And so this is to further exemplify that point. There are microbes everywhere. So whether or not they're on Mars already, if we try to colonize Mars, we will be bringing microbes along with us. So that brings me to the MinION sequencer. As opposed to a lot of other next-gen sequencing technology, such as the Illumina suite of products, we are able to get long reads using MinION. And here's the device right here. It fits in the palm of your hand, as opposed to Illumina machines, which easily can take up a table, or PacBio machines, which are easily taller and wider than I am, which would make them harder to bring into space. And so here's it being used on the space station. So no gravity and less gravity on Mars isn't a concern for the MinION because it runs on capillary action. And so here's some more on the MinION. what it's made of are these pores. And they pull the DNA strand through piece by piece. And as it flows through the pore, it makes an electronic squiggle. You can use that squiggle to then identify what base it was, whether it's A, T, G, C, or U. You can also tell methylated states, and they're working on doing non-DNA, non-RNA type nucleotides, such as Hachimoji, which was released in the last year. But one of the issues with this technology is it does have a high error rate of 1 to 10%. So with every advantage, there's also a drawback. Yeah, so they're really small pores, but you get these flow cells, you load it up, and you can run multiple samples all at once. So it is very handy, especially we're able to use these in remote areas while doing field testing, for example, on swine. So they're very portable and very easy to use. So my original project with the robotics team was to take soil, purify it on the rover, and then test it for DNA, or test it for nucleic acids, and then sequence it. So what I started was I got some dirt from one of the potted plants sitting around, autoclaved it to make sure everything in it was dead, and then added E. coli. I then purified the dirt sample to show that this can happen with environmental dirt, sequenced it using the MinION sequencer, and then assembled the genome. So the longer points here are the points where I had more coverage. So when you have a high coverage, especially with the MinION technologies, it kind of balances out the error rate. If you have 5,000 data points for one spot and it has a 10% error rate, you're pretty well able to separate out the errors. And then I do want to point out here, because it's easy to see, there is a gap at this point. The kind of genome I made with this was I was mapping back to a reference. And so that gap is most likely because I was using a lab strain of E. coli that just had that section of the DNA missing, most likely so it's not as pathogenic, and you can have people working on it in labs easier. So, the big project, Dinococcus Radiodurans. It is an extremophile. So, the way they found Dinococcus Radiodurans was they were trying to find a way to preserve canned food so that it would stay safe. They put it in the can, they hit it with radiation, and it's still spoiled. So they purified it and found out that there was the Deinococcus Radiodurans in it, because the radiation had killed everything else but this. So it's radiation resistant, and it's a 3.3 megabase genome, as opposed to E. coli, which was around 4.5 megabase genome. So it's a smaller one, which also makes it easier to handle as opposed to the human genome, which is magnitudes larger. So after, what I did was I cultured it in a lab, purified it, ran it on the MinION, and then you get all these squiggles. So there's a lot of programs you use to make the squiggles into letters and then take away the reads that they know are bad. So if there's over a 90% chance that it's wrong, you just cut it out. Or under a 90% chance that it's wrong, you just cut it out, you don't use it. And so this is the pipeline. One thing about using this in space would be this is a very computer-intensive process. And so I have to use the university's computer cluster to do this kind of work. So either the information would need to be sent from space back to Earth, or have a computer cluster on Mars so that this can be done there for whatever life detection we are doing there, and the health applications of being able to sequence in Mars. So in the last week, I was able to do the run and make a map similar to the one for E. coli. This area, that really thick blue part, has a read depth of over 5,000. That is really good. And then the small blue line is a read depth of 500, which is also good. I mean, full coverage of the genome, and this was able to be done in the lab. So what this means is that we got the genome. And as you can see, there was also, they're not as pronounced as on the other genome, but there are those little bits missing. Again, because I'm using a lab strain, I'm not going out and finding one naturally. But the next steps for this is to, well, the areas with the gaps, the reads that go over that area do go from one end to the other. So the next step is to use the reads I got back and have them mapped directly on top of each other, what we call a de novo assembly, as opposed to these mapping assemblies that I've done. This should be able to give me a reference genome, so once I start exposing these cultures to different kinds of radiation to see how they affect, I have a baseline that all these cultures came through that I can start to compare them to. And with that genome, it'll become a reference, so it's what I'll always compare to. I will also annotate it so that I can figure out what kind of genes these radiodurans are making. So the next step is to expose to the radiological stressors and see what genome changes occur. Some of the ones I'm looking at doing now are cold plasma, X-rays, and cosmic rays. I would expose the cultures, remove some of it, regrow it, and then in one generation test to see what kind of differences occur. Because Dinococcus Radiodurans must be able to do some kind of DNA repair, which would be how it survives the high radiation. So we are trying to see how does exposure to radiation change its genome. And then environmental stressors, including extreme temperature and desiccation as well, to see how it's changing the genome. And then in the long run, some other future experiments are to take those same cultures and see what of the transcriptome, proteome, and metabolome have also changed. That way we can hopefully find the web of proteins that are associated with the DNA repair that these Deinococcus Radiodurans are able to do. So, why does this matter? Well, again, when I started, when humans go to space, we will be taking our microbes with us. There is no separating us from the microbes that live in and within us and on us. And so we will need to see how they change, especially when it could change our digestive processes and how we absorb nutrients when we live in space. There's also the possibility that we'll run into space microbes. We'll be able to culture these while there, or without culturing like I did with the soil. Just take the soil, purify it, and culture it there without a need to send it back to Earth. But this could, in the future, possibly lead to medical treatments. If we understand how the Deinococcus Radiodurans are fixing their DNA, it is possible in the future we could try to make medicines that do the same, so that as people, plants, and animals continue to venture out into space, they won't have the same kind of DNA damage as they would without these possible future medical applications. Here's some acknowledgements to the groups that have been willing to fund me. So, thank you for listening. Yes?
Well, there's DNA repair, there's also DNA replacement, and Dr. Sinclair at Harvard has demonstrated that there's a backup genome. Are you considering that? Does your research consider that possibility that the backup genome that the Radiodurans is very efficient at bringing into play when it observes damage in the existing one?
Yes. So the way that the backup genome would most likely be stored as it is would be with multiple copies of a single gene, so that when one is damaged, it can overexpress the other one. Since we are only giving it one generation to proliferate after exposure, we should be able to see where the damage was done, and then if we follow up with the proteome and the transcriptome, see what they are expressing as opposed to baseline. So we should be able to see what is broken and where they might move it or what they might change to fix it.
Yep. And
we can also see what they will upregulate or downregulate to make up for any damage.
Is there a methyl clock? Is there a Horvath clock on the Deinococcus?
I am not sure.
So with like aluminum and stuff like that, you use primers to target certain regions on 16S, but I'm not too sure with the mini-ion and stuff like that, technically sort of how that works, because that also brings into considerations if you're targeting, say, like a quote alien life which uses the same sort of DNA we do, maybe it's panspermia, offshoot, distant cousin, whatever. How would you go about targeting things to get proper sequences out of it to actually work with these sort of general things?
So with the mini-ion, you don't need primers. You just need the nucleotide sequences, and then when you go through the library prep, you bind things onto them, but they're not primers. There's just more stuff that the pore will recognize and start to pull through. But because Illumina will cut up different parts with 150 base pair length as their general maximum length, as opposed to the, I think, 2,000 you can get with mini-ion for length reads. Basically, by using mini-ion, we don't have the bias of having to use primers, and we could possibly also use mini-ion for non-DNA or RNA nucleotides, which you wouldn't be able to do currently with Illumina.
If I can steal
Yeah.
one more question. What was your view, just more broad speaking? Because if we went to go back to, say, Viking with a targeted release, right? So we have things there. We got some yeses, some no's, some maybes, and it's a bit thing. When you have something like mini-ion, which has up to 10% error rate, you'd be worried that it could also bring in, say, another like labeled release fiasco, like Viking and stuff like that, going down in the future.
Yeah. So the 1 to 10% error rate while it's reading means it might confuse a C for a G. But at this point, if it was on a rover and went to Mars and it got any readings,
Yeah, okay, fair enough.
yeah. But the medical applications of if someone is in space and someone gets sick, being able to sequence whatever virus or pathogen, hopefully you're able to get a read depth deep enough to account, to make up for the percent error. So it also kind of comes down to how you culture. If you do find rare microbes on Mars, hopefully you can do like the soil project and filter or purify it and then do it, but you can also culture in between.
So it's a matter of biomass, essentially. The more biomass, the less error.
Okay, thank you everyone.